Good morning, everyone. Can you hear me?
Yes, we can.
Yes, we can.
All right, thank you. Welcome everyone to another exciting webinar where today we're going to focus on soil science. So to take us through, we have Ms. Dadirai Chinamo, a soil scientist with Kutsaga, who will take us through. Over to you, Dadirai.
Thank you so much, Tafazwa. Good morning, everyone. Welcome to this webinar. Morning. Welcome to this webinar, which is being hosted here at Kutsaga Research Institute, formerly known as the Tobacco Research Board. And for the next 30 minutes or so, we are going to have a talk on soil analysis for sustainable crop production. As introduced, my name is Dadirai Chinamo Chauruka. I'm a research officer at Kutsaga, and I specialize in research on soil fertility management, biofertilizer research, sustainable agriculture, regenerative agriculture, as well as climate change research. But today, my topic, soil analysis, will have its focus on soil fertility management. Before we delve deep into this presentation, we should ask ourselves why we should be concerned about nutrition. Is it important anywhere? To answer this question, I've chosen to bring the issue home and talk about humans first. So when you look at this guy, he seems off. And I tell you, malnutrition or lack of one essential nutrient can cause someone to actually feel sick, to be irritable, to feel tired all the time, an inability to concentrate. Some may even get into depression, lack of appetite, and above all, have a higher risk of contracting diseases. Well, in fact, some conditions are just a result of lack of one essential nutrient. We talk of kwashiorkor, just the lack of protein will cause someone to be that sick. So nutrition is very important. And ladies and gentlemen, we need to understand that the same scenario that happens in humans is almost similar to what happens to our crops in the field. With tobacco as an example, a lack of calcium causes deformed, curled or curbed leaves as shown there, leading to stunted growth and development. And when we talk of boron deficiency, this will result in malformed, distorted, or what we call rosette-shaped leaves that are very brittle and stiff, easy to break, and also death of the growing tip, just because of a lack of a small nutrient called boron. And when we talk of magnesium and iron deficiencies depicted there, they both show up as interveinal chlorosis, that is yellowing in between the leaves. But the only difference being that with magnesium deficiency, this happens on the older leaves, whilst with iron deficiency, the symptom appears on the younger leaves. And you should know that iron deficiency can be bad to an extent that the leaf can become nearly white, and there is drastic reduction in terms of yield just because of iron deficiency. And of course, we have our nitrogen deficiency, the small yellow leaves, the stunted growth, the low yield, and the poor quality, which some have actually experienced in the past season. And then we come to potassium deficiency, where there is chlorosis or yellowing of the tips of the leaf margins, which can progress inwardly, even to about a quarter or half of the leaf can be lost due to potassium deficiency. Though in some cases, this can correct itself during the season. So inevitably, with all these deficiencies, there is ultimately a decline in yield and quality to varying degrees, depending on the time or the stage of the growth the plant was when the deficiency started, and any corrective measures that were done but definitely you get a yield and quality decline when there is inadequate nutrition. So we talked about inadequate nutrition, but there are also challenges that come with overnutrition. For example, when we have high levels of nitrogen, we are still talking about tobacco. This can result in green tobacco at harvest and even after curing. It also results in increased disease incidences, fleshy yellow spots that will not cure well. I'm sure some of you have had such an experience where the crop is dark green during its vegetative stage. And then when we expect it to be ripening, it remains green and starts producing these fleshy yellow spots. So I think some of you have experienced that, and it fails to ripen. Again, you might get a problem of what we call fat stems that persist even after curing. They are very difficult to dry during curing, and this can be a source of pathogens. So overnutrition is also a challenge. So then, how do we know the right amount of lime in fertilizer to apply? We have seen that undernutrition is not good, as well as overnutrition. So how then can one know the right amount of fertilizers to use for a particular crop and in a particular field? Because our fields are different. This is where the issue of soil analysis comes into play. When we talk of soil analysis, it refers to the process of evaluating the physical, the chemical, and biological properties of soil to assess its fertility. So you see there that we are actually evaluating these three, the physical, the chemical, as well as the biological, so that we can get to a soil that is healthy. So when we test our soil, the first reason for doing the analysis is that we want to do soil fertility assessments. How fertile is your soil? Because we want to be precise about the amount of fertilizers to apply. We also want to assess the health of the soil by measuring things like your soil organic carbon level, your microbial activity, and in particular, especially when it comes to tobacco, your nematode assessments are very crucial, very important. Maybe not only in tobacco, but in some crops like your tomato, you need to know your levels of your nematodes before you get in with any chemical. And also we are testing our soils for environmental protection reasons. When we put the right amount of fertilizers, we are guarding ourselves against over-fertilization and against runoff, which might end up in our water bodies, in our water sources, our rivers, our dams, which can then cause eutrophication, algal blooms, dead zones in our rivers and in our dams. We have actually seen this happening on other farms where their dams have been contaminated to an extent where the water is no longer suitable for irrigation. So soil testing actually guards against this by allowing you to apply the amount of nutrients that are needed for that particular crop and for that particular season. Again, soil testing allows cost optimization, as it is a sure strategy to save money whilst still supporting plant growth. And when it comes to tobacco, we should know that tobacco is a hungry and nutrient-rich crop. You might appreciate that the amount of food that one eats is usually proportionate to the level of hunger that one has, well, under normal circumstances, of course. So you eat proportionate to the hunger that you have. So when we say tobacco is a very hungry crop, it means that it demands a lot of nutrients and it is exacting in its nutrient demands. For example, with our potassium, as long as it is available for uptake in the soil, the crop will continue to take it up, something that we call luxurious consumption, but leading to compromised yield and quality, and ultimately a decline in your profits. So it's very important that we are applying the amount of fertilizers or nutrients that are required. And when we are referring to the quality of the leaf, we are referring to both the physical and the chemical composition of the leaf. The physical being the ease of handling the leaf during harvesting, curing, grading, and baling. We have had some cases of increased scrap, what they call scrap marara mbani, during grading, or because of nutrient deficiencies that were encountered in the field. So we need to test our soils. And during the test, there is one important variable which is tested, which we call our soil pH. So the importance of soil pH is that it determines the availability of nutrients in the soil. So no matter how much fertilizer is applied during the season, if the soil pH is not right, those nutrients that you have applied will not be available to the plant, as most of them will be logged in the soil just because the soil pH is not right. As we can see from that graph that I've given there, the optimum pH for most crops is between 5.5 and 6.5. This is where we have most of our essential nutrients available to levels that plants can tolerate. And this is also the range where root growth is at its optimum. That is between pH of 5.5 and 6.5. On that note, we should note that most of our Zimbabwean soils are in the acidic range. And this means that our phosphorus, nitrogen, potassium, sulfur, calcium, they are all not available for uptake. But also there are other soils scenarios that we have started to receive where, especially on commercial farms, where the soil pH is now beyond 6.5, actually reaching to about 7. Again, you have other problems where you have your micronutrients being available to toxic levels, your manganese, your iron, your copper. You start experiencing problems of phytotoxicity because of the high soil pH. So this is very important for us to know. Now I want to move on to the steps involved in collecting a soil sample. I do hope we have understood the importance of having our soils analyzed before cropping, because the fertilizers that we use should always be based on soil analysis and never on general recommendations, as soils are different. So before you start collecting your soils, number one, you should know the timing of sampling. Soil sampling should be done after each cropping season. Why after each cropping season? We are saying during the season, there are changes that happen in the soil as the soil is very dynamic. During the season, these changes can be due to nutrient cycling, microbial activity, loss of nutrients to the environment, maybe due to leaching, due to volatilization. A lot happens during the season, as well as fertilizer generated acidity. For those who use ammonium nitrate, you know that there is some fertilizer generated acidity as the ammonium is being converted to the nitrate form, thereby lowering your soil pH. So for these reasons, sampling should be done after each cropping season before we get in with another crop to assess the current status of the soil and give correct recommendations. In the case of tobacco, this should be done soon after the rains, as at this time the soil will be stable. Number two, you need to divide your land into similar zones. We see from that picture there that there is that area where we have light soils and the area where we have dark soils. So that dark soils will be treated as a separate sample. You sample it separately, and the light soil is also sampled separately. You should not mix these, because definitely the nutrient composition is different. So the soil color is a giveaway. It can tell you that this land is different from this one, or this piece is different from the next piece. And also cropping history can also help you to divide the land into similar zones. Some of the lands, you know that although they have similar color, but in terms of crop growth, this land, there's always some lag in terms of crop growth as compared to the other piece. So you treat them differently, even according to your cropping history. After you've divided your land, you need to gather your sampling tools. The tools that we use for soil sampling, mainly we use what we call an auger. If it is accessible, it's the easiest tool to use, easy to use even in the field. If you don't have an auger, a spade will be the second best option. And lastly, if you don't have an auger, you don't have a spade, you can actually use a hoe. And also, you need to have clean containers to put your soil in. This should be clean because we want to avoid any contamination. So do not use any leftover sacks, fertilizer bags, unless you have thoroughly cleaned them, because we do not want to pick the fertilizer when we are doing our analysis. We assume that your land or your field is already fertile and you don't need much fertilizer. Yet it's the fertilizer from the bag that we have picked it during our analysis. So your container needs to be very clean. And you also need a marker, because we said we are dividing our land and separating into different lands. So for each land, it has to have a unique identifier. So you need to have a marker with you, and then you'll be writing the land in a manner that you will remember and you will know. And then when you get into the field, you need to have a sampling method. How are you going to sample? You're not just getting into the field to take just one sample, maybe from one corner or from the center. We're actually getting in the field to take many soil samples, and this should be in a non-sampling method. Why? Because we want to avoid bias. The soil samples that you are taking are going to be representing your whole field, and they have to be as representative of the field as possible. So that's why we advocate that we need to have a sampling method. There are different types of sampling methods that can be used: the zigzag, the random, the systematic, clustered. But for our tobacco farmers, for Kutsaga here, we advocate for the Z sampling method because we have seen that this is an easy method, whereas depicted in that picture there, you'll be just moving across the field in a Z manner in taking soil samples across the field as you move. And as you see, that area that is marked with an X is different in terms of color from the rest of the field. That's why I've marked it with an X, that you need to avoid such areas. So for you to have a representative sample, avoid different colored areas, avoid areas where you load off your fertilizers, areas where there are ant hills, you avoid. Areas where there are where there's dung, you also avoid. Because if you pick that area, it's like you are saying your whole field is full of dung and it's already fertile. And that's what we are going to pick. And those are the recommendations that we are going to give you. So it has to be as representative of the whole field as possible. And also to avoid bias and to make it more representative, you collect at least 10 subsamples per hectare. When I say at least 10, I'm saying the minimum is 10 per hectare, but you can go as far as your strength can carry you. Because the more samples you have, it means the more representative the soil is going to be for your field. Remember, when you get into the lab, after the sieving, the preparation of the sample, we are going to use maybe about just 2 grams of your soil to represent your whole hectare. So you have to take as many subsamples as possible. And you take them to a depth of 0 to 25 centimeters. I'm saying 0 to 25 centimeters, meaning that when you get into your field, you're not going to be scraping off any topsoil or anything that is on the top. You're just going to be taking right from 0 up to 25 centimeters. Some can actually reach up to 30 centimeters, but 25 centimeters will be okay. After taking these subsamples, you need to mix them thoroughly. So how we mix them as depicted there, we use what we call the quartering technique. You have taken your subsamples, you mix them in the sack or in the container thoroughly. You pour the soil sample on a clean sheet and you form like a circle. You form the soil into almost like a circle, and then you divide it into 4 quarters. That is the quartering technique. You divide it into 4 quarters. And then those 2 quarters that are facing each other, for example, in the picture there, that those that are in blue that I've written discard, you actually remove them from the sheet, you discard. And then you keep those that I've written keep. And then you mix them again. After mixing again, you do the quartering technique again, 4 quarters, discard the 2 that are facing each other and keep the other 2. So you continue this process until you get 2 kg of a soil. Because when you get into the field and you're taking many samples, when you come out, you'd be actually carrying a lot of soil. It will be more than a kg. But for analysis sake, for bringing for analysis, we just need a kg of the soil. So after thoroughly mixing, going over and over through the quartering technique, when you see that your soil is about a kg, that's when you stop, put in a clean bag, and bring for analysis. So this container that you are going to put should be well labeled. You are labeling where you have sampled, you are labeling the history of that land. You are giving your unique identifier, which you remember. So it's important to even map your own field and draw it on a paper and say, in this land, that's where I sampled and I labeled it land 2B. And then in this land, this is where I sampled and labeled land A, so that you remember when the soil results come out. So you put in a well-labeled container. We need, as I said, the date of sampling, your unique identifier, where it was sampled, and the analysis that is required. And it's important that we submit to an accredited laboratory. There are many labs that can do soil analysis, but it's very important that it has to be accredited. When we talk of accreditation, we are saying that this laboratory is under another board which sees to the issues of quality control. Quality control in terms of the machines that are going to be used, in terms of the analysis methods that are going to be used. Because, for example, if we just take phosphorus, there are different methods that can be used to extract phosphorus from the soil, but they do not all extract phosphorus to the level that we would require. And also it looks at even the person, the scientists who are going to be doing the extraction. So in an accredited lab, we ensure that you get the correct results for your analysis. Here at Kutsaga, we have an accreditation of ISO 17025, and also we do what we call proficiency tests, where we get blind samples that are being tested across different labs. And then from these blind samples, we actually analyze them, submit our results. So it's like an exam, we submit our results and we get our mark, and we know exactly where we are, exactly the results that we should have gotten and what how we did. So we continuously improve even in terms of our methods, in terms of our machines. So what I'm saying is you need to submit to an accredited laboratory. After submission, you get what we call a test report. This will now tell you a lot of things depending on what you've requested for. For example, here we are talking of your pH, your EC, the calcium, magnesium, right up to your phosphate. And you can also add your soil organic carbon, your sulfur, incubated nitrogen, and all those would then need to be interpreted by someone who knows how to interpret these figures. Because if we are going to give you, the farmer, these figures for you and say then how much fertilizer to put, you might not be able to do that. So these need to be interpreted by someone who's skilled and someone who knows how to do these things. So after this interpretation, you'll be given a recommendation. We pride ourselves here at Kutsaga because all our recommendations are based on research, real research that we have done in the field to say if you put this much fertilizer on this type of land, what will come out. So we do these recommendations. But what I want to urge farmers is we need to follow the given recommendations. It's very sad to see that a farmer has gone through all the steps of collecting the soil sample, submitting, paying for the sample, getting the recommendation. But at the end of the day, when they get the recommendation, then they look and they say, this fertilizer is too little, I should add more. Or this fertilizer is too much, I should add less. But you have done all that work for you to get the correct recommendation. So I want to urge farmers to follow the given recommendations and also to lime our soils. Because as I said before, most Zimbabwean soils are in the acidic range. So as a take-home message, I'm saying fertilizers are a costly input in crop production and must be efficiently used through a well-planned fertilization program based on soil analysis. It cannot be based on a general recommendation. It should be based on soil analysis. And number two, on a wise selection of nutrient sources. What we are going to give you is a guide in terms of the recommendation. But when you go out there, it's you who is going to choose which fertilizer source you're going to use from which company. But what I can tell you is from the trials that we have done here at Kutsaga on tobacco, in terms of different nutrient sources that are available on the market, especially the new fertilizers that are coming on the market, that are being sold as amendments, that are being sold as supplements. Most of them, we have found that they have no effect in terms of improving your yield and your quality of your tobacco. So we need to do wise selection of nutrient sources. With this, I want to thank you all for listening, and I'm happy to entertain any questions if you do have any questions for me. And those are our handles. You can contact us at those handles: Twitter, Facebook, Instagram, LinkedIn, and we have different recordings, even of other webinars that we have done on YouTube. Thank you so much.
Thank you so much, Dadirai, for the presentation. There you heard it, listeners and fellow growers, the importance of maintaining soil health in our soils as we aspire to achieve Are there any questions? Take this opportunity to for questions from the floor. Anyone questions? There is a question that we received on our chat. Craig Bondro, sending the graph is asking, is the pH graph in calcium chloride, potassium chloride?
Oh, okay, that one, thank you so much for that question. Yes, that one was on calcium chloride. And even when we do our soil pH analysis here at Kutsaga, we use the one for calcium chloride. It's more representative of what exactly takes place in the soil. Thank you.
Thanks, Dadirai. Any questions? We await questions from the floor. Any questions for Dadirai? Maybe while we're waiting for more questions, Nadira, I just wanted to, I was fascinated by your discussion on the various components for knowing what is required to ensure. So, in terms of the collection of Samsung, could you just maybe dwell a bit on some best practices that we can do in terms of collecting our subsamples, because we receive samples, growers tend to make the mistake of collecting from limited sources. But you talked about identifying the different areas in your field. If you could just comment on some best practices for collecting subsamples in the field.
Thank you so much. I see from the chat that there are people who are saying, oh, they can't hear the host. Am I audible?
You are audible, but the others are not.
Oh, okay. Thank you so much. I think there was a question there to ask how best can one collect a representative soil sample, because we have seen that some farmers bring in soil samples, and after the analysis, you can actually tell that there was a challenge in terms of the collection of the sample itself. So when you get into a field, it's very important that number one, as I said, we divide the land into similar soil. But during the sampling itself, you need to be careful in terms of the differences that will be within the field. We talk of the ant hills, we talk of anywhere where you see that it seems different, you should avoid those areas. Because we have seen other farmers, they actually pick fertilizer that was in the previous crop. So when you do, the moment you see that your sample or your soil sample that you have sampled, it is some trace of some fertilizers, you should not put it then in your container. You should actually discard it and take another soil sample. So that's the major mistake that farmers do, that they pick out some residual fertilizer that is in the field, and then that is what we pick during soil analysis, and then we say that your land is very fertile when it is not. So those are some of the things that you should watch out for. Thank you.
All right, thank you once again, Dadirai. I hope you can hear me in the audience. Can you just confirm that you can hear me?
Yes, we can hear you.
All right, thank you very much. Do we have any more questions for Dadirai before we round up? All right, let me just check the chats. I think there are indications of all right, I have a question from one of the viewers asking whether the webinar will be posted onto YouTube. Definitely the webinar will be posted on the Kutsaga account on YouTube. So please don't miss out if you joined the webinar late. Don't be disappointed. You will have access to the recording on YouTube. Let me at this point take the opportunity to thank all the listeners for staying tuned. We had a very insightful presentation on soil health and the different facets like undernutrition, overnutrition, why we test our soils, and also the important practices best practices for sampling, the timing of sampling. So I encourage all listeners to please stay tuned for the next exciting Kutsaga webinar. We have a number of webinars coming up. The next one entitled An Update on Breeding for PVY Resistance on Kutsaga Cultivars. Please don't miss this one. We do have a lot of leaf spot problems that we are likely to experience in the coming season with the predicted La Nina. So this is one not to miss. So at this opportunity, at this point, let me thank you all for listening to the webinar. Until we meet again, this is goodbye from me, your host, Tafadzwa Mahere. Please stay tuned for the next webinar. Thank you all.